Okay, let's look at some important information as well. What are the two types of nucleic acid? It's very familiar. It's DNA and RNA. What are the roles of nucleic acids? So you're very familiar with this one. So the DNA provides direction for its own replication, so it can replicate on its own. And then also very familiar because we talked about the central dogma of molecular biology. The DNA would be the one directing the production of the messenger RNA or the RNA, and then the RNA would be the one producing the protein. So very familiar. DNA produces the RNA produces the protein. And this is what we call as gene expression. The whole process from DNA to the production of a polypeptide or a protein is called gene expression. Okay, another important thing you have to remember, we're going now to the structures. What are the three parts of the nucleotide? Just remember three parts: the pentose sugar, the phosphate group, and the nitrogenous base. PPN, pentose sugar, phosphate group, and the nitrogenous base. So take note that these are the three important parts of the nucleotide. So we have here the phosphate group, which is connected to the fifth carbon of the pentose sugar. So take note, this is the carbon 1, carbon 2, carbon 3, and carbon 4. Take note, they are very important, and then carbon 5. The number of carbon or the carbon number is very important because the phosphate will be attached to the fifth carbon, and then the nitrogenous base, which is the third part of the nucleotide, is attached to the first carbon. So carbon 1 attached to the nitrogenous base, carbon 5 attached to the phosphate, and another important carbon number is carbon number 3 over here, because this is where the other nucleotides will be attaching themselves to produce a polynucleotide or the nucleic acid polymer. We'll talk about that a little bit later. Now, just a side note, take note that you have OH here and OH here. What kind of pentose sugar is this? This is called a ribose because it has oxygen on both of this carbon 2 and carbon 3. But if you have DNA deoxyribose, one of this will disappear. So you will only have one oxygen at this point. Okay, making it more simple, you have the base here, the deoxyribose sugar here, and the phosphate. And two more things you have to remember would be there that the bonds between the phosphate and the sugar, and the bond between the sugar and the base. First of all, you call this as the phosphodiester bond, while you call this one as the glycosidic bond. Okay, so those are two bonds you have to remember. Okay, let's look at the components of the nucleic acid. Again, when you have a series of nucleotides together, you call that as a polynucleotide. It is a polymer. And each polynucleotide, as mentioned, is made up of a nucleotide over here. So take note that this is the nucleotide, the phosphate, the sugar, and the pentose. But if you remove the phosphate, you will now call the sugar and the base simply as nucleoside. One more time, the whole thing, three of them is nucleotide, while these two would be the nucleoside. Okay, what are the bases of the DNA? Another thing that you have to take note of. So for the DNA, there are four bases you have to remember: adenine, cytosine, guanine, and thymine. ACGT. Okay, so those are the bases of DNA. However, how about RNA? In RNA, you still have the same bases except that you will change one of them. Adenine, cytosine, guanine, which is the same with the DNA. However, you will change thymine into uracil, and that will give you the RNA bases, which implies that if you see a nucleic acid with U's, letter U, in it, this will tell us that this is an RNA. Whether it is double-stranded or single-stranded, it doesn't matter. It is RNA once you find a uracil there. But if you find a nucleic acid that would be containing thymine instead, then you will be very sure, whether it's single-stranded or double-stranded, doesn't matter. When you see a thymine, it is automatically a DNA. Okay, so another thing you have to take note of, what are the classes of the bases? Well, take note of the first one, which is given by the mnemonic purga, because the first of the class is called purines, and the two bases that will be included in the purines, as given by the mnemonics purga, is the G and the A, which would be the guanine and the adenine. So that will help you remember which are the purines. It is the guanine and the adenine. And which this will be leaving us with the last one, the second class, pyrimidines. And the clue here is that all bases with letter Y will be under the class of pyrimidines. So you have here the cytosine and the thymine. And since uracil is also a uracil is also a nucleotide, uracil is also a base, then you also have to include this, and this is actually included in the pyrimidines. So to remember the pyrimidines, just remember CUT or CUT, it is inclusive of cytosine, thymine, and uracil, and that will be pyrimidines. And this is what pyrimidines and the purines look like. So the adenine and the guanine are actually double ringed. Here's a five ring and a six ring put together. That's adenine and guanine. And then you have a single ring for thymine, cytosine, and uracil. So this would be the pyrimidines, and this would be the purines. Now, please do remember something very, very important, that the oxyribose is the sugar for DNA, and the ribose is the sugar for RNA. As mentioned a while ago, ribose has both oxygen present in the second and the third carbon, while the oxyribose, one of the oxygen is removed there. So de means none, so one of these would be removed. Now, there is what we call as the base pair rule, and this is very, very important, especially later on as we talk about replication, because the thymine or the uracil will always be pairing with adenine, and the cytosine will always be pairing with guanine. Okay, and that will be very important. So C is always pairing with G, and A is always pairing with T, or A is always pairing with U when we talk about the RNA. Now, it's very important also that you take note of the percentages. So, for example, if you have 5 cytosine, automatically you will know that there will be 5 guanine. If there are 203 thymines and you have a double-stranded DNA, automatically you know that there are also 203 adenine or adenine present. If you have 500 uracil present, you already know that if the RNA is double-stranded, then you're very sure that you will have 500 as well adenine, if that is double-stranded. Okay, so if it's single-stranded, it doesn't matter. So take note of this one. These are what we call a base pair rule. Thymine always binding to adenine, uracil always binding to adenine, especially for the part for transcription. Cytosine binds with guanine. Okay, so that are the things that we have to take note of. Again, very important here is the highlight that the 5 carbon is connected to the phosphate, and the 3 carbon that exposed here is very important, because if you will be producing a polynucleotide, the phosphate group of the second nucleotide needs to form a bond, which is a phosphodiester bond, with the 3rd carbon, leaving this 3rd carbon open, exposed, which allows another nucleotide to form a bond there, a phosphodiester bond again, and then so on and so forth. So take note of this one, very important, the 5' N and the 3' N. 5' because the 5 carbon is here, 3' N because the 3 carbon is the one exposed over here. So the production or the connection or the dehydration reaction producing the bond to produce the polynucleotide is always 5' towards the 3' direction. Okay, so these are RNA single-stranded, the DNA is double-stranded, and this is just a very comical way of showing the DNA RNA. So DNA is this one, RNA is the single-stranded over here. And let's just finish up with by finishing up this by making a comparison. So what's the sugar of DNA and RNA? The sugar would be deoxyribose. For DNA, RNA, ribose. What are the bases? We're very familiar. DNA, adenine, guanine, thymine, and cytosine, while RNA is adenine, guanine, uracil, and cytosine, uracil being the one different for the RNA. How many strands for DNA? You have double-stranded with base pairing. How about RNA? You have single-stranded. Helical? Yes for DNA. Helical? No for RNA. That ends our discussion. See you in the next